Okay, so heredity. Genetics is the study of inheritance of certain characteristics due to the passing down of genetic material. At O levels, we're looking at mono, one, hybrid inheritance, meaning that characteristic is only controlled by one gene. Let's say I have the gene for eye color. Maybe I have different kinds of gene. I have the allele for blue color, allele for brown color. So different forms of genes are called alleles. Can? I try another way. A gene is a segment of DNA. So you occupy a certain position called locus. Okay? Gene is a small segment of DNA. We already learned this. Okay? Different form of the same gene is called alleles. For example, I have the gene for can roll tongue or not. Then I have the allele for can roll tongue, cannot roll tongue. Or I have the gene for height. So I have the allele for tall or allele for short, quoting for. So the letters represent the different alleles. Homologous. Homologous chromosome means same. What do I mean? Same shape, same genes, same size. We have 23 pairs of chromosome in our human body. 23 pairs means 46. One set was from mom, maternal. One set was from dad, paternal. Okay, so this is trying to show that, hey, actually we have 46, 23 pairs. Some terms to clarify. Phenotype, PH, physical, is usually a physical trait, observable. Okay, and this can be influenced by not just the genes, but also the environment. For genotype, is at the genetic level, the genetic makeup for the organism. With that, we introduce terms like homozygous. Big T, same. Small t, same. So long, same is homozygous. Heterozygous is whereby the trait is different, one big, one small. Then we talk about the term dominant and recessive. Dominant allele. The moment I have one of the allele, the trait will express itself. Recessive, I will need both the allele to be present in order for the trait to express itself. So example here, I have a homozygous dominant, homozygous recessive, heterozygous. What I see, purple color, white color, that's a phenotype. What is the genetic makeup, the genotype? Big A, homozygous dominant, small a, or big A, small a. These are called the genotype. Now, when we talk about crosses, okay, this is two, this is showing two. So actually, you only need half usually. What do I mean? It usually stops here. Let's just do the bottom, we'll do, let's just do this one. Okay, so from here, we always start with what we can see. Phenotype, genotype, gamete, F1 generation, genotype, F1 generation, phenotype. Okay, if this is too confusing, let me just type it out for you. I repeat, I always ask parental, what's the phenotype? Then, what's the genotype? I make sure I circle the gametes. Show the cross. From the cross, I have the genotype of the offspring.
Appearing on the screen.
Sorry, Shannon?
There's nothing appearing on the screen.
Thank you. Is this better now?
No, we're still kind of stuck on the slides and it's like empty.
So there's nothing on the slides.
Okay, I'm sorry for the Wi-Fi connection. Is this better? No. I've been typing.
No, yeah.
No, okay, I'll restart everything. Give me a while. So I was saying the genetic diagram. I would always write down the phenotype of parents, so parental phenotype. With that, I have the parental genotype. And then the gamete circle, you do the cross, Then you have the offspring, also sometimes called F1 generation, the genotype. Obviously, from the genotype, you can determine the phenotype of the offspring, and then the phenotypic ratio. Then I always have a concluding statement of the ratio. This part, okay? So this is the genetic diagram. I want to give you an example of this. And the computer is trying to handle how much we are doing right now. All right, we do whatever we can. Okay. Okay. So with this, let's try. Okay. Let the allele for big B be dominant. Be the dominant allele for, let's say, brown eyes. Okay. Let the small b be the recessive allele. And therefore, it will code for, let's say, blue eyes. Okay. I have both brown-eyed parents, brown-eyed parents. How do I even get a blue-eyed child? So firstly, I was saying that, my parents are brown eyes. Short form brown eye. So brown and brown. The only way it's that they are both heterozygous. So I'm going to circle the gametes. And then I'll do the cross. For me, I like to keep this simple. First with second, second with first, second with second. Big B, small b, big B, small b, small b, small b. And that is the genotype of the offspring. So phenotype will be brown, 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 blue. And because of that, I have a 3:1 ratio. If the question asks for probability, I will say that, hey, there's a 1/4 or 25% chance of the child being blue-eyed. So that is the basis for the genetic diagram. If it comes out, yay, it can be 5 to 6 marks or 3 to 4. It depends on the year. Save this for later. Okay. Of course, if it's just a Punnett square, you can use the Punnett square for working. So if it's just an MCQ or they don't need you to show how you derive your answer, so these are Punnett squares that you can use. Still remember to circle the gametes though. Okay, so the gametes, right? So I want to talk about something called test cross, yeah? So how do I know whether the parent was a homozygous dominant or was it a heterozygous? I can do this by crossing it with a homozygous recessive. From the ratio, I can tell whether the original was homozygous dominant or heterozygous. How do I know? Okay, for example, this plant looks tall. So is this tall plant big T or big T small t? I can only find out by crossing with a short plant, which is small t, homozygous recessive. Then I realized, oh, I planted the first batch. 100, all 100 became tall. Oh, the parent was homozygous dominant. However, if I did the cross, right? So out of 100, maybe like 74, 3/4 were tall. Then I can realize and conclude that, hey, the original parent was heterozygous. So that is called the test cross. Okay. This is a test cross, and I'm use it to determine the original genotype of the organism. So this was what I just went through. Big T, small t. Is there an error here? Big T, big T, big T. Yeah, I say wrongly. Give me a try. Let me work out my working out. Yeah. There's an error here. This is hybrid test cross. Okay. I need to replay this. I'm sorry. If it's a big T with small t, it would be 1:1 ratio. My bad. Okay, I copy the Punnett square over. Okay, so big T, small t, big T, small t, small t, small t, small t, small t. So it should be a 1:1 ratio.